think one of the most important developments that we're seeing now in whole genome sequencing is the development of long-read sequencing and its ability to be used for analysis of whole genomes. Traditionally, we've been limited to the traditional 2x150 paired end sequencing type of a model where it's very good at calling point mutations and very localized phenomena, but not quite as good at identifying deletions and translocations that really are the underpinning of a lot of the use cases for whole genome sequencing. So I think long-read sequencing is really going to change our ability to do that. I think one of the major problems that we're going to come up against in the coming years as we try to use some of these genomic technologies in clinical use is the ability to manage the volumes of data and the amount of computing power that we need to manage and to analyze the large volumes of data. And I think that's really going to necessitate a move towards the implementation of smaller focused panels of deep sequencing data, moving away from whole genome sequencing types of studies of large cohorts. Once we've learned what the key mutations are, particularly in diseases like cancer, where there are going to be a limited subset of mutations that are actually driving those diseases, I think that once we know that and we can move toward a focused panel, the implications of the amount of data and the ability to handle those data in a rapid amount of time will be paradigm shifting. I would say again, I think whole genome and targeted long-read sequencing is going to be a major disruptor. I think the ability to do direct RNA sequencing with some of these technologies is going to change things quite a bit compared to what we can do now. It's going to give us the ability, particularly when married with traditional shorter read sequencing technologies, it's going to give us the ability to have insights on the structural variation that underlies a lot of disease-associated genomes that we just would not have had before. So I think that those types of technologies have the potential to really be disruptive. So I hope that the future of NGS in 2019 and beyond is really a move away from the discovery phase into the translation phase. I think that we've learned an enormous amount about the way genomes are structured, the mutations that underlie risk of disease, underlie pathogenicity of disease, underlie outcomes for disease, and we've really now got to move toward, and the onus is really on us, to move toward a model whereby we're starting to implement some of these changes and really look for tests that implement whole genome sequencing as well as some other targeted sequencing approaches to really drive clinical implementation and make it matter for people.